Hi everyone, and welcome to our Biology Daily Booster. What we're going to be doing today is number 6 in our series for paper 2, and it is advanced listed information for both the higher and foundation biology folks. So relevant for all of us today. What we're going to do is start off by understanding how we can work out some of these genetic crosses, because they quite often ask you to work out the genetic cross or the offspring that may occur from certain parents. So what I've given you here are 6 steps to success, basically. This is going to ensure that whatever the marks are, you will get all of them. So first thing, state the phenotype of both parents. Now remember, the phenotype is kind of what you see as the easiest thing there to remember it as. So that would be brown eyes, blue eyes, etc. The genotype are the genes that are present. So that's our step 2, state the genotype of both parents. Step 3, state the gametes for each parent. Draw the Punnett square. Then we're going to state the phenotype of each offspring in the Punnett square. And finally, we state the percentage of each genotype present in the offspring. So those are the 6 steps we need to actually succeed in this question and get max marks every single time. So if we have a little look at this question, then 2 people who are heterozygous for retinitis pigmentosa are expecting a baby. Draw a genetic diagram to calculate the probability that the baby will have the condition. Use capital R for the normal allele and lowercase r for the retinitis pigmentosa allele. So this is a 3-mark question, folks. Now, first thing, heterozygous. Break it down. Hetero means different, zygous is meaning alleles. So what we have then in our parents, they are not going to have retinitis pigmentosa, okay? Because this, in the earlier part of the question, was said to be a recessive condition. So what we know as far as the parents go then, so we will have the mum is just going to be capital R, lowercase r. Dad will also be capital R, lowercase r. So when we set out our little Punnett square, it's basically like the old-fashioned windows used to draw at primary school with a little bit extending out each side. So what we're going to do, I'll put Dad on this side, Mum up the top. Now, we've got a capital R and a lowercase r. So if I put a capital R there, I'm actually going to do these in different colours so we can see. Lowercase r, same for Dad, 1 capital R, 1 lowercase r. Now, all we're going to do is, starting with the capital R for Mum, whatever's at the top of that column, fill it in to the 2 squares beneath. Then, same for Mum on the other side, whatever's at the top of the column, fill it into the 2 squares underneath. To Dad now, whatever's at the start of the row, put it in the 2 squares to the right, and then the same for the other one, put it in 2 squares to the right. So now what we need to do is identify whether they've got the condition or not. So 2 capitals, that one is going to be normal. 1 capital, 1 lowercase, normal, capital and lowercase, normal, and then 2 lowercases, that means they've got the condition. So they have the condition there. So what we can see is we have 3 normals and 1 with the condition. So it's a 3-to-1 ratio, but we're asked to calculate the probability. That would work, 3-to-1, okay? We could also say that it's going to be a 1 in 4 chance, or we could write that as a 25% chance, 0.25 chance. Any of those will be acceptable answers for this, because what we're really looking at there is that we've all given probabilities in just different formats, but all of them would be correct. But that's all we need to do to get these genetic diagrams right every time. The next thing they could ask us to draw a genetic diagram for is sex determination. Now, in females and males, we need to know the chromosomes present. In males, 1X, 1Y, females, 2 X's. Please go careful how you write this, because I've seen this come up as questions in the past. And if they say which sex chromosomes are present in females, and you just write X just a single X, it's going to be marked wrong because there are two X's. Just like with the males, don't just write the letter Y, you need the X and the Y. So make sure that if they ask you, you use both chromosomes, XY for male, XX for female. If we wanted to show how that was inherited, very simply, you would have the female, the male, and then all you do is just fill it in, just like we did before. And in that case, it will always be 50% chance of either gender. Another word we need to understand the meaning of is the word mutation. So if they ask you to define or explain what mutation is, then it's just a change in the base sequence of the DNA. Now, mutations occur spontaneously in DNA replication, but the chance of mutation will increase if you're exposed to certain things. And two things to remember: ionizing radiation, such as UV, and certain chemicals like ethanol and benzene. So generally speaking, the more you're actually exposed to those things, the more likely you are to have mutations. As we've already said, that genetic variation comes down to obviously differences in that genome. If we've actually got different versions of alleles arising from mutations in the DNA, we get greater genetic variation. So mutations can actually be beneficial. However, not all of them are beneficial. Some of them will have no effect on the phenotype whatsoever. That may be a mutation that still codes for the same amino acid, therefore changes nothing. They may actually influence the phenotype, so there may be one of these that just gives a slight alteration but doesn't altogether change it, or it could literally determine the phenotype, and that's how we get our variation in a species. And depending on what the mutation is, it may be harmful, beneficial, or just neutral, have no survival benefit either way. Examples of some harmful mutations then. Cancer, most likely one. Okay, so if you're asked about a sort of risky or dangerous mutation, cancer is the best one to go with, which is just where our cells are growing and dividing in an uncontrolled manner. We've got another example with cystic fibrosis, where we've got a channel protein that's been created in an abnormal way, so it doesn't work properly, and therefore we get the thick, sticky mucus. And in sickle cell anemia, we get a different shaped protein in the red blood cells, so they are actually a different shape altogether. Neutral mutations, ones that will have zero effect on your survival either way. Your ability to roll the tongue, whether you can roll your tongue or not, it's not going to affect whether you're going to survive or not. And whether your earlobes are attached to the side of your head or kind of just free floating. Beneficial mutations for bacteria, that antibiotic resistance, which we'll look at more in Unit 6, that is beneficial to them, not so much for us. Skin colour can be very beneficial there, because if we consider the fact that people with much darker skin colours are less likely to get skin cancer, quite a benefit there. And the FOXP2 gene, which actually gave us speech in the first place. What we need to understand for the higher folks is basically how this occurs. So what are these mutations? How do they actually occur, and what effect do they have? In terms of DNA itself, what we have are coding sections, which are our genes, and then they're separated from one another by this little section of non-coding DNA. So what we actually find is that if you get a mutation in the coding section, obviously that could affect the protein made. If the mutation occurs in the non-coding section, it won't change the protein whatsoever. So depending on where the mutation is going to be part of whether it has a significant effect or not on the organism. As for how these mutations happen, there are three key ways this is going to happen. The base will either be swapped out for a different one, so individual base swapping. We can add a base, or we can delete a base. In terms of the effect that each of those has, if we just swap an individual base, so if we have a look here, we can see our sequence. We started with a T in the original, and it's been swapped for a G. So when we consider our little triplet codons that we use obviously to code for the amino acids to make our protein, we can see that once we've actually divided them up, the only one that's changed is the first one. because it was ATA, it's now AGA. So that could be a different amino acid, but it's only a single amino acid that changes. The rest remains the same. If, however, we add a base, then what we can see is we've actually changed all of those codons from that point on. So where we've added that additional T, that first codon has changed, then the second one, the third one, and so on. So this has a much larger effect. Same with deleting. If we've deleted a single base, it's going to change all of the triplet codons from that point forwards. So adding base, deleting a base, they have much greater impacts than just changing an individual base. The way that we then see the impact of those changes to the triplet codes is that we're then going to have a different amino acid. If the amino acid sequence changes, then the protein folding can change, or we can make completely wrong proteins altogether. This is where they can start to draw on some of that paper 1 knowledge. If we think back to paper 1, one of the key things we focused on was enzymes. Hopefully we remember the key bit about enzymes is that they are proteins, and when we look at the shape of our enzyme itself, it has this region here, which is a very specific shape called the active site. Now the shape of the active site is determined by the sequence of amino acids. So if we have a mutation which has led to a change in the amino acid sequence, the shape of the active site could also be completely different. At that point, we're no longer going to have an active site that's complementary to our substrate. It won't work. So watch out for these bits that do have the links to the paper 1 content, and be prepared to bring that knowledge onto this second paper. Last little bit to consider here then is the fact that in terms of how we actually know where to start actually copying a gene from, we have these things called start codons. So it's literally the codon of 3 bases that literally says start here. Now those are located in the non-coding section of DNA. So what we find is if the mutation occurs in that start codon, then the gene may not be transcribed full stop. So if that's happened and we're not able to transcribe the gene, we clearly won't make that protein at all. So whatever that protein did is not going to happen in the organism. And obviously that can be incredibly serious, potentially life-threatening. Don't forget that if there are some bits you want to go over in a bit more detail, then have a look at the video on the main channel. You can also use your revision guides, your classwork notes, and of course I would suggest looking at some past exam papers just to see the way that they phrase these questions. And then don't forget to join us again tomorrow for our next Biology Daily Booster.